Good evening, everyone. It's probably a good time for us to get started. Hi, my name is Tom Ulrich. I'm the Associate Director for Science Communications here at the Broad Institute, and it's my pleasure to welcome everybody in the room here and everybody watching on YouTube to the second in our Broad at 15 talk series. The Broad, as you know, came into being in 2004, and since then we've seen an amazing pace of progress in many fields of biology and medicine. These talks are to celebrate that progress and also the parts that the Broad has played in it. It's my pleasure tonight to introduce our speaker, Dr. Todd Golub. Todd is our Chief Scientific Officer here at the Broad. He's the Director of our Cancer Program, also of the Gerstner Center for Cancer Diagnostics here at the Broad, and he's also a Professor of Pediatrics at Harvard Medical School. Todd's going to talk to us tonight about cancer precision medicine, what that phrase means, the progress that has been made towards that goal over the last 15 years, and what's yet to come. A couple of other small things before Todd comes on. If you have any questions for him after his talk, please raise your hand for one of the mic runners, or there'll be a number on the screen where you can text your messages, or text message your questions in as well. And if you're watching on YouTube, you can also use YouTube chat. And if you're tweeting, please use the hashtag #BroadAt15. And without further ado, Todd Golub.
Thank you, Tom. Good evening, everyone. Thanks for everyone who fought the Kendall Square traffic to get here in the thick of rush hour. I'm really excited to have a chance to talk to you about what's happening in the field of cancer precision medicine, what we mean by that, what's happening at the Broad, celebrating our 15th anniversary, but the bigger picture also of what's happening in the rest of the world in precision medicine now, and how did we get here? Before I get started, I want just a couple of quick disclosures. I've been a consultant to a number of companies that are listed here, but I'm not talking about any of their products today. So what do we mean by cancer precision medicine? What does a vision for this look like? The way I think about this is that it means that for all patients everywhere, genomic information from the patient's tumor will guide the selection of optimal targeted therapy. That is, that we move away from the idea of giving a collection of poisons, chemotherapy, same kind of treatment for all patients, but instead give targeted therapies to the right patient at the right time. This isn't exactly a new idea, but it's really gaining steam today, and that's what I'm going to explain to you today. To try to put this in a little bit more context, I want to frame this in one of the challenges and obvious opportunities that I faced as an early pediatric oncologist a number of years ago. Described two patients. The first I'll call Charlotte. She was a 6-year-old diagnosed with acute lymphoblastic leukemia, we call ALL. She received standard chemotherapy and immediately went into remission. 30 days later, we did a bone marrow test, no signs of leukemia in her bone marrow. She was in complete remission and remains alive and well today. Second patient, diagnosed the same year, John, was diagnosed also with ALL, what we thought was the same disease. At age 7, got the standard combination chemotherapy, but didn't respond at all and didn't respond even to even stronger chemotherapy and a bone marrow transplantation, and unfortunately died the year later. So this is the challenge. How do we understand this dramatically different response to treatment for what seemed to be the same disease? Clearly, these two kids didn't have the same disease, and this is what we need to understand. So what I'm going to try to describe today is not just where we are today on the 15th anniversary of the Broad Institute, but a bit of history around how we got here. And I'm going to go back not just a few years, but actually a lot of years. Starting about 1600 BC, which is the earliest recorded notion of the idea of cancer. This is good and actually quite amazing that on this Egyptian papyrus, the Edwin Smith papyrus, there was the idea of cancer as a tumorous growth. The treatment wasn't all that helpful. A couple of magic spells were prescribed. I assume they didn't work all that well. But it would take a long time, 3,500 years, for that magic to start to turn to mechanism. And that's what I want to describe today. I think maybe the clearest example of this is a local one, Sidney Farber, for whom the Dana-Farber Cancer Institute was named here in Boston, was the first to show in 1947 that if you gave what we now think of as a kind of chemotherapy to kids with ALL, like the ones I described before, that at least some of the patients would have an initial response to therapy. This was quite spectacular because prior to this, the feeling was, well, once you have bad cancer that's spread throughout the body, there's really not much you can do if you can't cut it out with surgery. But Sidney Farber showed that you could get initial responses to treatment. Unfortunately, those patients all relapsed and developed resistant disease. And so he and his colleagues over the next 30 years reasoned, well, maybe if some chemotherapy is good, maybe more chemotherapy is even better. Let's try combinations of different chemotherapy drugs, many of which didn't work any better than just the one alone, some of which were not tolerated because they were too toxic. But then by about 1990, it was clear, 1980, sorry, it was clear that a certain combination of chemotherapy could induce not only initial remissions of kids with ALL, but some kids could even be cured. And now basically using that same kind of approach, about 80% of children with ALL can be cured with combination chemotherapy. So some chemotherapy good, more chemotherapy looked like is even better. And that's good in a way, but it's kind of not heading in the direction of what we now think about as cancer precision medicine, piling on more and more nonspecific drugs. So about the same time that this combination chemo idea was gaining traction with ALL, a different scientist in Shanghai, Zhu Chen, actually was inspired by a Chinese philosopher from 500 BC, Confucius, and this is true. Zhu Chen, this is at the end of the Cultural Revolution, was reflecting on the teachings of Confucius, and I'll read this quote. If you use laws to direct the people and punishments to control them, they will merely try to evade the laws and will have no sense of shame. And he equated this with these laws and punishments being like chemotherapy. Okay? But if by virtue you guide them and by the rights you control them, there will be a sense of shame and of right. And so he had this idea that maybe there would be alternatives to just piling on the chemotherapy. Maybe you could induce cancers to do what you want them to do if you did this in a more thoughtful, gentle way. And at the same time, 1980, he read a paper published in the Proceedings of the National Academy of Sciences, written by a group at the National Cancer Institute, who found that in a particular type of leukemia, not ALL, a different subtype of leukemia called acute promyelocytic leukemia, or APL, that at least in a petri dish, if you took those leukemia cells from the patient and treated them with a naturally occurring hormone called retinoic acid, it's a little bit hard to see in these old photomicrographs, that kind of as Confucius was suggesting, you could gently ease these leukemia cells to become less angry and proliferating and induce them to become more mature, at least in the petri dish. And so based on this intersection of philosophy and some petri dish experiments, Zhu Chen said, all right, let's try a clinical trial of this idea, only in these patients with APL, acute promyelocytic leukemia. So here's the compound called cis-retinoic acid. It's a particular type of retinoic acid that was reported in that paper. Zhu Chen is in Shanghai in the mid-1980s, and he couldn't get this chemical, but he could get something related, something called all-trans retinoic acid. And you can see it just differs by a little bit at the end. And he said, that's close enough. Let's try that, okay? So he tried it in a patient. Now, 1985, a 5-year-old girl with acute promyelocytic leukemia, not ALL, this particular subtype, gave her the drug. She had not responded to any chemotherapy prior to this. Treated with Atra, complete response, and I'm told that this girl is still alive today. This then started a proper clinical trial, initially small, 24 patients who got this drug, Atra, the one by mistake, not cis-retinoic acid, which it turns out actually doesn't work as well in patients. So he lucked out. 23 out of the 24 patients had a clinical response, and this is now the standard of care for patients with this subtype of leukemia. So almost overnight, this molecularly defined what we now know is a molecularly defined subtype of leukemia went from the worst possible kind of leukemia to have to now the best kind of leukemia to have, and it's treated with a treatment regimen with retinoic acid, this hormone, as a background. It would take about a decade after Zhu Chen's initial observation to figure out why this is working, to turn this magic of what the hormone does to an actual mechanistic understanding. And it was discovered that there's a mutation in the gene for the retinoic acid receptor. It's the protein to which this hormone, retinoic acid, Atra, binds, and that explains why patients respond. But they only respond if they have this particular mutation. That's the early beginnings of cancer precision medicine. But this is sort of done in a weird way, right? Because the first observation was that of a clinical response, but no idea what explained it. It was kind of a phenomenon, and they worked backwards to figure out why. Could you go the other direction? Could you start with a molecular understanding of a cancer, and from that infer what a good treatment might be? So that happened actually starting in the late 1950s. Peter Knoll and colleagues working in Philadelphia made this observation that there's a funny-looking chromosome shown there. It was very low resolution look at the genome at that time. You remember your genome was packaged into chromosomes. And he said there's something funny about this part of the chromosome. He couldn't even tell which chromosome it was. But he said, every time a patient has this particular type of leukemia, they've got this funny extra band here. Maybe that screwed up chromosome actually causes the leukemia, and if we could really understand that molecularly, that could become a therapy, and maybe that's how we treat these types of patients. The community didn't exactly embrace that idea because many people said, oh no, you've got it backwards. It's actually that you get cancer, you get leukemia, and when you get leukemia, your chromosomes get screwed up afterwards. And so this is just irrelevant. And so chasing that mechanism isn't going to be a path to a cure. And it wasn't clear what was going on here, so they just called it the Philadelphia chromosome. And it remained that way for 10 or 15 years until Janet Rowley, working at the University of Chicago in 1973, discovered that funny-looking Philadelphia chromosome was actually part of chromosome 9 stuck onto part of chromosome 22, what we call a 922 translocation. And then another decade later, researchers in a couple of groups discovered the exact genes that were broken by that chromosome translocation. Half a gene on chromosome 9 called ABL, and half a gene on chromosome 22 called BCR, making this BCR-ABL fusion. And then, years later, scientists figured out what does this BCR-ABL fusion protein do? There's still this question of, well, maybe this occurred after the patients get leukemia, and it's just, it just happens. It's not really causal. And we would want the new treatments for cancer to be targeting the root cause of disease. So then, right in the building next door at the Whitehead Institute, George Daly, who's now the dean of Harvard Medical School, but then was a postdoc, in 1990 did the critical experiment. He took this BCR-ABL mutant gene and he put it in the bone marrow of a mouse. And he waited. And the mice got leukemia, just like the humans did. So this proved that this BCR-ABL, the product of this funny Philadelphia chromosome, actually caused leukemia. This was critical for the early foundations of cancer genomics to show that really cancer is a genetic disease and you can work this out. It's not overly complex. You can do it. It took a long time. And then Brian Drucker, following that observation, said, well, we need to make a drug against this thing. Particularly targeted this overactive ABL gene that is activated by virtue of its fusion to BCR. He had to go from company to company to company to get anyone interested in doing this because it just wasn't the way he thought about cancer treatment, which was mostly focused on chemotherapy at the time. But finally, he got some traction in a company that's now Novartis, and they made a drug against this thing, and they did the clinical experiment. Could you treat patients, first doing the genetic test to see whether the leukemias had this Philadelphia chromosome 922 translocation, BCR-ABL fusion, treat only those patients with the drug, would they respond? And the answer was pretty clear. Made the cover of Time magazine around 2001, where essentially all the patients responded. And this is now the standard of care for these patients. So here, starting from the genetic discovery of what was going on in these leukemias, working all the way through the mechanism to make a drug and then deploy that drug only in those patients. Now, there were those that said, oh, but it's a great cancer drug, a great leukemia drug. Never mind this precision medicine stuff. Let's just try it in other patients with other kinds of leukemia and other kinds of cancer. Doesn't work. Precision medicine really works. It only works in the molecularly defined patients where it's intended. So this is very exciting. At the same time, there were a number of concerns where people said, well, maybe this is really just a rare example. Maybe it's really the only example of this kind of response. Cancer is really complex, and it took 40 years to go from that discovery of the Philadelphia chromosome to a drug. We can't really imagine doing that over and over again. How could we think about doing this systematically? And this is where I think the Broad comes into play. So this was now around 2004, launch of the Broad Institute, where the concept here was to say, can we make systematic our discovery of these kinds of cancer genes and other genes that are associated with disease, because they will inform the future of therapeutics. Now, this is an interesting time, 2004, because the human genome had just been sequenced, in large measure right here, actually in a building about 3 blocks away, in a Budweiser and popcorn distribution facility for Fenway Park. But all 3 billion bases of the human genome as part of the International Human Genome Project were decoded. It only took 15 years and about $3 billion to do the Human Genome Project. To give you a sense of what's the size of 3 billion letters of the genome, if you were to print them out the size of the font of New York Times newsprint, and then laid out that newsprint from here in Kendall Square, it would take you all the way to Providence. So that's a lot of letters. And so the notion that you could, if you were interested in discovering other cancer-causing genes, it wasn't even a debate. It was unthinkable. There was no discussion about, oh, we'll just sequence the whole genome in a tumor to discover new things. It was so off the radar. But there were a couple of scientists, a couple of Brodies, Matthew Meyerson and Bill Sellers. They said, well, we can't do the whole genome, but we got to start someplace. Let's start with what was at the time the largest cancer genome project ever done, which kind of in retrospect seems sort of comical because it's so small. But they said, well, let's take 50 genes out of the 20,000 in the human genome. Let's start with 50. That's less than 1% of the human genome. And let's sequence it in tumors from, turns out to be a collection of 58 tumors, lung cancers, from patients with lung cancer, and see do we see any mutations in any of those 50 genes that are in the tumor that aren't in the normal cells from those patients, because those would be candidates for new mutations like BCR-ABL that cause, in this case, lung cancer. So it took a long time to do this project, but they did it and discovered mostly that all of the 50 genes, 49 of them didn't show anything interesting, but one of them, in about 10% of the patients, had a mutation in a gene called EGFR, the epidermal growth factor receptor. This was really exciting, not just because there was a new cancer gene discovered, because there happened to be a drug that was being developed that was intended to inhibit EGFR, a so-called EGFR inhibitor. And so they asked the question, oh, well, maybe it's the patients who have this mutation that will be the ones that respond to this EGFR drug. And that's exactly what happened. If you have the mutation, you respond to the drug. If you don't have the mutation, you don't respond to the drug. And this is now standard of care for any patient anywhere with lung cancer should get an EGFR test. And if you have the mutation, you should get an EGFR inhibitor. If you don't, it's clear that you don't benefit at all. In fact, you do worse if you get that drug compared to chemotherapy. So this was a real turning point because this really sent the clear signal to the whole field that BCR-ABL and Gleevec, that drug that I showed you, wasn't just a curiosity, wasn't a one-off. There were going to be other examples like this. And if we could find one such important cancer-causing gene and a therapeutic match for it by just sequencing 50 of all of the 20,000 genes in the genome, we ought to think about how to make it possible to sequence all genes. And so this gave inspiration to another international project, this time not the Human Genome Project, which was to sequence a first reference normal human genome, but an international cancer genome project. The one in the US is called the Cancer Genome Atlas, an international one, the International Cancer Genome Consortium, to try to make it possible to resequence all the genes in all common cancers. And this has now happened. And just to give you a spectacular sense of how genome sequencing technologies has changed over the years, this is just the work at the Broad Institute sequencing now whole genomes, all 3 billion bases, not this little less than 1%, 0.1% thing, but the whole thing. 136,803 as of last count in June, genome sequenced just at the Broad. And it's happening all the way, all around the world. A new whole genome coming off these sequencers every 6 minutes. The first one took 15 years. So this is really changing how we think about just about everything. We now can say with confidence, we actually know what are all the common cancer genes. Of course, there are some uncommon ones in very rare cancers or rare mutations in even some common cancers, but we now can say with confidence there are about 400 common cancer-causing genes, and we should be thinking about those and how to approach those in a precision medicine kind of way. And increasingly, it's becoming possible for patients to get this kind of testing, either in major cancer centers or in companies doing this type of testing. Now, one challenge with this cancer genome testing is that it has to start with a biopsy of the tumor. And this works in many cases, but in some cases, patients aren't able to get a biopsy. Sometimes there's a patient is diagnosed with cancer, they get treatment, the tumor comes back, and the doctors say, well, we know what it is. We know the cancer's come back. There's not really a clear need for another biopsy. We know your old cancer's come back. Or maybe the tumor is in a dangerous location in the body, and doing a surgical procedure or even a needle biopsy might be too risky and certainly uncomfortable. What if you could make it possible to decode the cancer genome in a non-invasive kind of way? And so here's where scientists at the Broad and elsewhere have been thinking about what we now call blood biopsy. What if it was possible to actually analyze the cancer genome, a tumor buried somewhere deep in the body, just from a tube of blood? This is something that I thought was never going to work. It was a cool idea to think that maybe there would be little fragments of DNA from the tumor floating around in the blood, and somehow you could capture them and sequence them. Turns out this actually works, that you can actually reconstruct the entirety of the tumor genome, not the normal cells in the body, but the tumor genome from the tiny fragments of DNA that can be collected in just a tube of blood. This, I think, is going to have pretty significant impact in a number of ways. We think it's going to become possible to molecularly monitor response to treatment using these kinds of blood biopsy tests. So in addition to a CAT scan or an MRI, where, yes, you can see if the tumor is shrinking on therapy, and are you on the right track, or do you need to change treatment course? Blood biopsy will make it possible to get molecular information of what's happening to the genome as the patient is undergoing treatment, and do this with exquisite sensitivity. You can also imagine that this could begin to turn our healthcare system into more of a learning system. What if this kind of cancer genome analysis wasn't something that just happened at research universities or at the Broad Institute or a major cancer center, but if any patient, anywhere, could go to their local pharmacy or have an Uber, have a phlebotomist arrive in an Uber, which exists, and have the phlebotomist take a tube of blood, send it into a lab, and get cancer genome information. And what if everybody could then have a mechanism to share their information? Could we really accelerate the pace at which we make these connections between particular mutations and particular responses to therapy? And so this, in part, inspired another large project anchored at the Broad that we call Count Me In, led by Nick Wagley and Corey Painter here at the Broad, where the goal of this project is to make it possible for any patient anywhere in the world to share their data. So at the moment, you may know that if you go to a major cancer center, you can enroll in a research study to share your genomic information and your clinical information with the research community. It's possible to do that, but most patients aren't treated at major medical centers. It should be possible for any patient to raise their hand and say, I wish to share my information with the research community to accelerate the pace of discoveries. And this project makes it possible to do that. So patients can go online and sign consent, informed consent online, because privacy concerns are very significant, and we want to make sure that people understand what the data will be used for and what will not be used for. So patients can agree to participate in a research study. And the availability of blood biopsy is now making it possible to have it much easier for patients to donate their genomic information to studies such as Count Me In. And a little hard, doesn't project so well here, but we now have thousands of patients from all 50 states in the country that are participating in this direct-to-patient research study, which is new and, I think, very exciting. Now, is it enough to just know what all these mutations are? Probably not. What you'd really like to know is, what are all the genes in the genome that are required for a tumor cell to survive? Because if you knew that, you'd have a complete roadmap for what drugs need to be developed in the first place. Some of them might be mutated and might cause cancer. Others of them might just be an Achilles heel, a vulnerability in particular types of cancer that could be exploited as an anti-cancer therapy. But which genes are Achilles heels in which cancers, and how could you possibly know? You could make a drug for each one and test them, but that would take, hundreds of years and an enormous number of clinical trials, so that's not going to work. What if you could use genetics to turn off each? There are only 20,000 genes in the genome. What if you could turn off each of them one at a time and figure out whether, when you turn off a gene, does the tumor cell die, or does the tumor cell not care? So this was just sort of an idea until new technologies became developed, and you may have heard of one of them called CRISPR, which is this really amazing system that can go in with a molecular scissors and seek out a particular region of the genome that researchers specified and say, cut here and only here. So you can design one of these CRISPR reagents, one for every gene, have it cut that gene, inactivate it, and then observe the cancer cell and see, did the tumor cell survive or not? This actually works. So it's now possible, using CRISPR technologies and related technologies, to eliminate each gene, 1 through 20,000, each in a row, and observe, did the cell live or die? And of course, we want to do this not just with a generic cancer cell line, for example, but with cancer cells that represent the diversity of human cancer. So this is still a project that's ongoing. It's something called the DEP map, or dependency map, the discovery of all of the genes in the genome that different cancers are dependent upon for their survival. We've done this for about 800 human cancers so far, testing all 20,000 genes in each of the 800 cell lines. So this is about 16 million data points generated in one project, all made available without any restriction to the research community, so that we now will soon have a complete map of what are the Achilles heels of all the common human cancer cells. This is testing this genetically, which is really foundational, but because it provides a blueprint for what are the important drugs to make for the future. What about existing drugs? Could you make a map of which tumor types are sensitive to which drugs? A lot of drugs have been developed. Some for cancer, most not for cancer. Well, the standard way to do this is to take a cell line representing a particular type of cancer and treat it with a drug and see what happens. And this is shown here in a part of a small petri dish where when their cells are alive, they look dark purple. And if you treat with a drug that is effective at killing the cancer cells, they appear kind of white. So here's an example of a drug that happens to kill that particular cell line. But again, that's a tiny bit of data. We want to have this comprehensively across what we now know to be the molecular heterogeneity of cancer, and not just a drug, but how about all drugs? Like all drugs ever approved by the FDA for any reason. So we actually did that experiment using some new technology developed at the Broad, where we could insert tiny molecular barcodes into each different cell line, say 500, or in this case 578 different human cancers, insert a barcode, all done in the laboratory, not a clinical trial, and treat them with over 4,000 drugs. So in one experiment, 2 million data points of which tumor is sensitive to which drug. And this is really, we're still in the middle of analyzing these data, but it's pretty spectacular to be able to have a map like this. We never had maps like this for cancer, and we can see already, for example, that there are unexpected findings. Drugs that were developed for arthritis or for high blood pressure, for diabetes, are selectively killing subsets of tumor types. Not all cancers. That actually wouldn't be the direction we want to go. That's more like going back to chemotherapy. But they selectively kill subsets of cancers, and we're now working up what's the mechanism by which they do that. But having these kinds of comprehensive, systematic maps give us the starting point for the discovery of potentially new uses of old drugs or new therapeutic strategies that could make for future new drugs. And like with the genetic CRISPR data, we give this all away. So we just released this data set of a couple million drug sensitivity measurements on the internet for the research community to use. Meanwhile, while all this is happening, with the focus on tumor cells, there's a quiet group of scientists, in particular Jim Allison, then at Sloan Kettering, now at MD Anderson, who was saying, you know, you guys are focusing too much just on the tumor cells. If you actually look at a tumor under the microscope, it's not just a ball of tumor cells. There are other cell types in there too, like blood vessels and fibroblasts and immune cells, lymphocytes, for example, maybe it would be possible to get the immune system to attack tumor cells. What was the response to that kind of idea? He and his small band of colleagues were largely ignored. Actually, not even ignored. There was disdain. That was such a crazy idea, couldn't possibly work. Why were they wasting taxpayer money pursuing this line of research? And as many of you have probably heard, this actually turns out to work spectacularly well. That there are molecular switches in these immune cells, particular types of lymphocytes like T cells, T lymphocytes is shown here. These red guys are T cells that are attacking these larger tumor cells that are shown in gray. And you can make drugs to activate these T cells in order to help them recognize the tumor cells that they should have been recognizing as foreign in the body to begin with. And in some patients, but not all, there's an amazing response to this new group of drugs, and in a number of cases, cures. They look like real cures. Now, is this an alternative idea for cancer precision medicine? No. In fact, it's almost a poster child for the idea of cancer precision medicine. Because it turns out that just about a year and a half ago, an immuno-oncology, so-called IO drug, was the first ever of any type to be approved by the FDA for use in any tumor type. He said, we don't care, actually. Is it lung cancer, breast cancer, whatever kind of cancer, if you have a particular genetic aberration called microsatellite instability, it doesn't matter what that means. But you make a molecular measurement of the genome, and if you've got that, this drug was shown in clinical trials will work, and the FDA said, yes, that's okay. So I think what this signals is a real turning point, because it shows you that the idea of targeted therapy and immunotherapy all come together around this precision medicine idea. And it also, I think, starts to indicate what's going to slowly happen over time, which is that we're going to have to rewrite all the textbooks of oncology, which at the moment are organized based on the organ in which the tumor arises. If it shows up in the lung, it's a lung cancer. If it shows up in the breast, it's a breast cancer. And what I think these kinds of precision medicine laboratory experiments and now clinical experiments are teaching us is that what's even more important than anatomy are the molecular underpinnings of cancer. And so that we need to rewrite the textbooks in molecular terms. What are the mutations and other causes of cancer? And we need to classify tumors based on that. And we need to get really good at measuring these molecular aberrations, mutations and other abnormalities in the tumors, and use that to guide the development of new therapies and to deploy them first in clinical trials and then in routine clinical practice. So to end, let me just summarize a little bit about what I said and where I think this is all heading. In the not too distant future, it's going to be the reality that all patients will have their tumor genomes analyzed at the time of diagnosis, and then later again on treatment. I think there's an interesting set of questions around this. You could say, well, this is advanced 21st century technology. Is this going to be technology available only to the wealthiest patients in the wealthiest country? Or is this going to equalize access to best diagnosis and the appropriate treatment? I happen to think that, at least over time, it's going to level the playing field. And it will mean that any patient anywhere can get to the right diagnosis with a simple genetic test without the need for an extensively trained expert pathologist to get the diagnosis. Not everyone agrees with that, but I think that's the way it's going to go. We're going to see, because the data are just too voluminous, rather than looking at the mutations and then trying to read the tea leaves, we're going to see increasingly machine learning algorithms saying, all right, here's the totality of the data, and here are the predictions of which drugs are most likely to work. Will, I think, increasingly get traction as we go forward. As more and more data are generated, shared in the research community, these algorithms will be able to be trained to recognize, when I see this series of mutations, it means that this is going to be the set of Achilles heels, and those Achilles heels will mean these are the drugs that will work. That's the world we're going to move to. As I mentioned before, blood biopsy is going to make it possible to monitor response to treatment in a much more accurate and molecularly defined way than we can now. It's going to become increasingly possible for patients to share their data if they want to. At the moment, we have this crazy system where most of our clinical information is locked up in our hospital medical records, and who knows how you could get it out if you wish to share it with the research community. We need to make it possible, and it will happen, for patients to say, count me in, I want to share my data, I'm going to press this button, and it's going to share my data, certain aspects of which I may wish to share, certain aspects of which I may not want to share, may not want to share at all. The patients should have that decision, not the hospitals, not the doctors, not the insurers. I think that's a world we need to move towards, and that's a world we will move towards. We'll see a new crop of targeted drugs that will be developed based on these new kinds of cancer maps that I described. It will still be critical to test these in clinical trials because laboratory research is not the same as clinical research, but I believe we'll see success rates of clinical trials go up substantially because we can know so much more about how drugs work and which patients are the right patients to get them before the first clinical trial even opens. So I think the success rate will increase, and we're already starting to see that happening for oncology drugs that are developed in this cancer precision medicine kind of mode. Drug resistance will still be a challenge, just like you've probably heard about antibiotic resistance being a problem for bacterial infection, tuberculosis. It's kind of the same challenge with cancer. This is going to be a problem that we need to figure out. Combinations of targeted therapies will be the solution, but we're going to have to get through that period of figuring out how to overcome resistance, not in a magical way, but in a mechanistic way based on the understanding of the disease. So I think you've probably already gotten the sense, I hope, that this is going to be a challenge, but this is a definable challenge, and eventually cancer will yield to this kind of approach. I do not think that, despite the fact that today maybe it's only a minority of patients that benefit from this idea of cancer precision medicine, I don't think this is a fad that's just going to go away. It's here for good. And I would say, the next generation, or maybe the generation after that, it's going to be the routine where all patients, of course, get treated in this kind of way, and it's going to substantially change the landscape of how we think about cancer, I hope, forever. Thank you very much for listening. I'm happy to take questions.
Thank you very much, Todd. And the floor is open. The mic runners are running, so if you have a question, go ahead and raise your hand, and they'll get the mic to you. While we're hunting down the first people... Oh.
How big a problem do you see as genetic heterogeneity to all these studies progressing successfully?
Yeah, I didn't quite see where that came from. So genetic heterogeneity is significant. It turns out that if you look inside any individual tumor from just a single patient, there's a lot of molecular heterogeneity within the tumor. Some of the cells within the tumor might have one mutation. Others, cells within the tumor might have a different set of mutations. And we need to understand that. So I think what's becoming already clear is that the best targets for treatment are going to be those mutations and other molecular abnormalities that are present in all of the tumor cells. Those tend to be the ones that occur earliest in the development of a cancer. You think about cancer, it's sort of like evolution sped up really fast. Not over millions of years, but over, let's say, the decade over which a tumor arises. And over that decade where the tumor is developing, it's sprouting in other directions with heterogeneity as it evolves. I think if we tried to go after each of those, what we call subclones, the individual microcosms, within each tumor, that's going to be really tough. But if we go after the root of the cancer, go after those changes that cause the tumor, all of the tumor cells to proliferate and survive, I think that's going to be a better strategy. But that's also what combination therapies are going to have to address, this heterogeneity challenge.
You got a question over on this side?
Hi, I'm over here. So I actually work in neuroscience and neurotherapeutics, and so I don't work in the cancer field at all. So I guess I'm wondering, like, what lessons do you feel like this field of cancer precision medicine, what lessons do you feel like are generalizable to other fields of medicine? Because I think about in psychiatry, there really isn't a really good molecular understanding of a lot of diseases. We're really just starting to come to that. So do you see an applicability of some of the tools and approaches that this field of cancer precision medicine has yielded for other fields to take into account?
Yeah, that's a great question. I think there are approaches that can be transferred. I think the whole notion that we could develop a molecular classification of disease is a very powerful and general concept. So rather than describing cancers based on the part of the body in which they arise, we describe them molecularly. Rather than describing other diseases like psychiatric diseases based on the symptoms that the patients have, we could describe those diseases in molecular characteristics. Now, it's a little bit trickier in neuropsychiatric diseases. At least in cancer, we know which are the affected cells. It's the cancer cells, and we can take them out of the body and study them. For other diseases, more complex interaction between brain cells and immune cells and other types of cells. But I think the concept of a molecular classification of disease, not trying to have a blanket treatment for all patients, and then also molecularly monitoring response to intervention in the lab, response to intervention in the clinic, I think those principles do extend.
Saw a couple of hands up down this way, but I guess there's a question up there.
Thank you for this awesome series, firstly. So I work for Shepherd Therapeutics. It's a rare cancer setup, and more often than not, we see that the cell lines that we are working from, the molecular signatures of that differ vastly from the patient tumors. And recently, Broad came up with an algorithm which it actually distinguished between driver mutations and passenger mutations. Could you give us a brief on how you do that work, and where are you between cell lines and the discrepancies that we see within patient samples? Thank you again.
Yeah, so that's a little bit of an inside baseball question. I'll try to translate it a little bit. So in order to do experiments in the lab, what we typically do is take, for example, from a biopsy, or if a patient is having a tumor removed entirely, take a bit of those tumor cells from the patient and grow the cells in the lab, and then use that as a model for the type of cancer the patient had. But sometimes the molecular details, the mutations in the tumor cells, aren't exactly the same as what's in the patient's tumor. And we just had this discussion around molecular heterogeneity. If there are different subclones within the tumor, each of which have distinct molecular profiles, maybe one of them will grow out in the lab and the other one is tested, let's say in the clinical setting. And so sometimes there can be a disconnect there. I think what that means is that we need to use these experimental models as good models for whatever genetics are represented in that particular model, rather than saying this is sort of an avatar, the cell line being an avatar for the patient. And so if I can test things in the cell line avatar, maybe that will tell me what to give to that patient from whom the cell line was derived. There's some differences of opinion there, but I think we're just starting to get a handle on how different are the cell lines from the primary tumors? How much difference matters? Maybe they're different, but it doesn't really matter in an important way. Maybe it matters in a trivial way, and we shouldn't be too distracted about it. But it's something we need to be on top of.
Todd, I want to jump in with a question that came in through the text number. Do you foresee a day when things like DepMap testing or the drug sensitivity testing you were talking about could become standard of care for patients?
Yeah. So that actually connects to this past question. So the way we're currently thinking about this is that we're going to do these laboratory experiments to learn all the rules. Every time I see these mutations, it means these are the Achilles heels, and those are the drugs that work. And then, having learned all those rules, then when I see those same mutations in a patient, I can say, we've seen that before. We saw that in the DepMap. We saw it in the lab, and we can then infer, all right, here's the right set of drugs for that patient to get. That's what I'm most optimistic about. An alternative view would be to say, well, maybe you don't need this prediction. Maybe what we should just figure out how to do is, when a patient is diagnosed, take a sample of their tumor, bring it directly into the lab, and test it with different drugs, for example, and see which ones work. And if a drug works, give that drug. There are some experiments to do that, and in some cases, maybe that's even feasible. It's technically very challenging to generalize, I would say, and I can't currently think of a way, for example, to test tens or hundreds or thousands of drugs on a tiny little biopsy. But this is what we're going to have to figure out.
Over on the side.
Hi, thank you. There's been a lot of advances recently in things like wearable devices where, for example, you could monitor a patient's temperature during a clinical trial so they don't need to come in for a physician's visit. I was wondering, do you see wearable devices or point-of-care devices helping to advance precision medicine? And are there any initiatives supporting either of those things at the Broad that you'd be able to speak to?
Yeah,
Thank
I think
you.
wearables are really exciting. When you think about, in the context of a clinical trial for any disease, what happens? You get a drug, you come in occasionally to see your doctor, who then records, how was your past couple of months since I last saw you? Do you feel better? Did you tolerate the drug well? Were you able to move around a lot? If you could actually collect that kind of information with more precision and just more data, I think that's all going to get better. I suspect that the value for wearables will not be in oncology first, but in other areas, and certainly, there's a lot of interest in watches that could monitor your heart rate and test for arrhythmias, for example, in a continuous sort of way. That's going to be a very powerful way to try to understand who's at risk for various types of cardiac disorders. So I think we're going to see more and more wearables being used in the context of clinical trials to inform what's actually happening. And also, even after drugs are approved for use, we need to get better about collecting more and more information just from their general use in the population. And I think wearables will help with that as well.
So my question, thank you for a very interesting talk, is two parts. The first is on blood biopsies. Since the cancer cells are sloughing off part of their individual cells or parts of their DNA, other cells throughout our other organs are probably doing the same. How easy is it, and what's the basic technique for separating out the cells that you want to really sequence? And the second question I have is, what are the possible prospects near and far down the road for then modifying the genes remaining in the human after the tumors have been killed, since those tumors arose from other cells, ordinary cells in the body that may still harbor some defects?
Yeah. Okay, two really interesting questions. So first, the idea of blood biopsies. There are two ways you can think about blood biopsy. One is to try to capture an intact tumor cell that's floating through the blood. And there are ways to do that. They're really rare, like less than 1 in a million cells in the bloodstream. Daniel Haber at Mass General Hospital has been a pioneer in doing this. Those cells are few and far between, but you can find them. The other approach that I was really focused on here isn't so much looking for intact tumor cells, but looking at tiny fragments, just a couple of hundred letters at a time, where the DNA is floating around in the blood. And here, the fact that we know that cancer is a disease of the genome and that tumor cells acquire all these mutations, it's the presence of those mutations that allows us to distinguish a fragment of DNA that came from a tumor, chock full of mutations, versus a fragment of DNA that came from just normal organs in the body. So that's the basis of making that distinction.
But do you sequence all the cells in the blood to find which ones are which?
We sequence all of the small fragments of DNA in the blood, yes. And the majority of them, in many cases, are normal. Well, that's okay. DNA sequencing has gotten so cheap, it doesn't matter. We can just hunt for the mutant ones and count how frequent they are. And in particular, we think it's going to be possible to see, even if it's a small fraction of that DNA that is mutant and therefore coming from a tumor, if that fraction starts to grow over time, that's a signal tumor's starting to come back. And our hypothesis, not yet proven, is that if we could start to see that growing and intervene early, heading the wrong direction before there's a big mass on a CAT scan or an MRI, that there would be better clinical outcomes. But that needs to be tested in a clinical trial. Second question, I think, reflects the idea that tumors just don't go from nothing to something in one fell swoop. There's a gradual accumulation of molecular aberrations over sometimes decades. And it's now clear that our bodies are full of normal appearing cells that also have certain cancer-related mutations, but they don't have the full complement, the full set of mutations necessary to make a cancer. They've got maybe all but one or all but two, and that's kind of how it works. So I think an interesting idea for the future around preventing cancer, which of course that's what we'd ultimately like to do, not to just treat it, with precision therapy, but to prevent it in the first place, would be in part to use genetics and exposure histories to identify patients at risk for developing cancer and try to cut off this development of cancer, this multi-step progression of cancer before it's a full-blown tumor. And there are ideas about how that might happen. That's going to be a longer project, but I think that would be very exciting. If, maybe it'll turn out every year on your birthday, depending on what risk of cancer, type of cancer you're at risk for, you take a dose of a medicine which then nips those precancers in the bud and reduces the population of cells that are at risk of becoming full-blown cancers. That would be exciting, not yet feasible today, but someday I think that will be possible.
Todd, I want to jump in with one more question that's been texted in. We've talked a lot tonight about genes and mutations in cancer. Can you have cancer without mutations in genes?
Yeah, so it is complicated, and I think there was an earlier question about this idea of passenger and driver mutations. So it turns out, and I told you about this controversy of, well, that funny-looking chromosome, does that cause the cancer, or do cancers form and then the chromosomes get screwed up as a result? It actually turns out both are a little bit correct. It's clear that mutations do cause cancer, but then sometimes what happens is that the genome really does get messed up, and that messed up genome results in a scattering of mutations across the genome, many of which don't really matter. They're just a signal of having a messed up genome. In some cases, if you have a mutation in the process that repairs your DNA when it gets damaged, let's say from sunlight, if you have a mutation in that gene, then you acquire more mutations, most of which don't cause cancer, the rare ones do. So we're trying to figure out which are the ones that are really causal, which ones are just there to begin with. So far, there aren't examples of cancers that have no mutations. They may exist, but they're pretty rare. There are probably other mechanisms that collaborate with mutations. The so-called epigenome is probably important. The immune system is probably going to be important. In some patients, a tumor may arise because there are many mutations, but a strong immune system. In another patient, there might be fewer mutations, but an immune system that is less good at recognizing and killing the cancer. So I think it's not going to be as simple as just counting mutations. But so far, all tumors that have been examined have at least one.
And I think we have time for one more question from the room. Somebody has one of the microphones in the middle.
Yes, thank you for the talk. And
Yes, I
you
just can't see
actually
where you are. Okay, got it.
you actually quite answered my question, because I wanted to ask you what you thought about prevention versus cure of cancer.
Yeah.
And I was actually wanted to know if you had more details about prevention and where it's happening, this prevention efforts. And so, yeah, I was thinking, could we use blood biopsy, for example, as a cheap testing of cancer, for example, to see the tumor fraction in the blood? Or in an ideal world, we could even think about building a vaccine against cancer through enhancement of the repair mechanism of the DNA. So do you envision a department at the Broad who could do something like this?
So those are all both great ideas, and those kinds of experiments are ongoing in a number of places around the world. I would say I'm not so optimistic about cancer prevention that just prevents all cancer in all patients, at least to start. What I am optimistic about is using, again, this idea of precision medicine and using a genetic understanding of disease to identify patients that are at particular risk for developing a cancer, and then saying, okay, in that population of patients that are at high risk of getting, let's say, breast cancer, is there an intervention that could be done in that patient population? So an example of that is, for example, BRCA1 or BRCA2 mutations that put women who have those mutations at risk for developing breast cancer. There is an intervention, mastectomy, preventively, to prevent breast cancer, very effective. Will there be more elegant preventive strategies than that? I think so in the future, but I use it to illustrate the point that that kind of intervention is best done when you can identify a patient population at risk. In individuals that are smokers or have a smoking history, they're at higher risk of lung cancer. So that's going to be the place to first test lung cancer prevention strategies, therapies, for example, vaccines or other non-vaccine kind of strategies. There are groups in one company in particular that is testing the idea. Maybe you could use blood biopsy to screen the general population to identify patients that have mutant DNA in their blood and therefore might have cancer and not know it. That's really attractive in principle. I'm less optimistic about that strategy working, at least initially, in an unselected population, because if you think about it at a population level, if the test isn't completely accurate, then you're going down a diagnostic path of CAT scans and MRIs and biopsies, maybe for a false positive, which even if it's rare at a population level, winds up being a lot of intervention that may not be useful. So I think these ideas are all really good, but should start in patients that are at particular risk of developing cancer or other diseases.
All right. Thank you very much, Todd. Everybody, Todd Golub.